Stages in B-cell development are distinguished by the expression of immunoglobulin chains and particular cell surface proteins. The stages in primary B-cell development are defined by the sequential rearrangement and expression of heavy and light chain immunoglobulin genes. The first classification of B lineage cells into different developmental stages was made according to whether they expressed no immunoglobulin chains or the immunoglobulin heavy chain only, or both heavy and light chains. Subsequently, intermediate differentiation stages have been distinguished on the basis of the expression of other cell surface proteins together with direct DNA analysis of the state of the immunoglobulin gene loci. The earliest B lineage cells are known as pro-B cells, as they are progenitor cells with limited self-renewal capacity. They are derived from pluripotent hematopoietic stem cells and are identified by the appearance of cell surface proteins characteristic of early B lineage cells. Rearrangement of the immunoglobulin heavy chain locus takes place in pro-B cells. DH to JH joining at the early pro-B cell stage is followed by VH to DJH joining at the late pro-B cell stage. Productive VDJH joining leads to the expression of an intact mu heavy chain, which is the hallmark of the next main stage of development, the pre-B cell stage. The mu chain in large pre-B cells is expressed intracellularly and possibly in small amounts at the cell surface, in combination with a surrogate light chain to form the pre-B cell receptor. Expression of the pre-B cell receptor signals the cell to halt heavy chain locus rearrangement and production of the surrogate light chain, and to divide several times before giving rise to small pre-B cells in which light chain rearrangements begin. Once a light chain gene is assembled and a complete immunoglobulin M molecule is expressed on the cell surface, the cell is defined as an immature B cell. The expression of the immunoglobulin heavy and light chains are key milestones in the differentiation pathway. These events do more than simply delineate stages of the pathway. The expression of an intact heavy chain and later of a complete immunoglobulin molecule actively regulates progression from one stage to the next. All development up to this point has taken place in the bone marrow and is independent of antigen. Immature B cells now undergo selection for self-tolerance and subsequently for the ability to survive in the peripheral lymphoid tissues. B cells that survive in the periphery undergo further differentiation to become mature B cells that express immunoglobulin D in addition to immunoglobulin M. These cells, also called naive B cells until they encounter their specific antigen, recirculate through peripheral lymphoid tissues where they may encounter and be activated by the appropriate foreign antigen. And that stages in B cell development are distinguished by the expression of immunoglobulin chains and particular cell surface proteins. Have a great day.